regarding meiosis 2. So in the meiosis 2, excuse me, so in this you can see the meiosis. So in the meiosis, it is mainly divided into, so the major part will be the prophase, G1, S and G2. Remember, G2 is mainly meant for protein synthesis. G1, the cell will be increased in its size and as well as metabolically active. At the same time, it also produces endoplasmic reticulum, Golgi bodies, ribosomes, except these mitochondria and chloroplasts, remaining all are developed. And now we are dividing this into, we have the M phase. So we have two things, meiosis 1 and meiosis 2. And this we call it as interkinesis. Now let's see, this again we have, this is meiosis 1. This is meiosis 2. Right? This is meiosis 1 and this is meiosis 2. In the meiosis 1, we have M phase as well as cytokinesis. And here we have prophase. The prophase is divided into 5 phases. Leptotene, zygotene, pachytene, diplotene, diakinesis, and we have this metaphase 1. Anaphase 1 and telophase 1. Right? Here also we have very simple prophase 2 because it is meiosis 2, right? Metaphase 2, anaphase 2, and we have this telophase 2, and here we have the cytokinesis. So let's see about meiosis 1. The meiosis 1, the prophase is the first meiotic phase. Okay, is typically longer and more complex when compared to prophase of mitosis. So it has been further subdivided into following 5 phases. So it is typically longer and more complex. Mitosis prophase, here the meiosis prophase is more complex. So we have leptotene, means thin threads, zygotene, paired threads, pachytene, thick threads, diplotene, 2 threads, and diakinesis, terminalization and or passing through or dividing the through. So let's see leptotene. In this stage, the chromosomes become gradually visible. The chromatin gradually visible, even under light microscope. The compaction of chromosomes continues during the leptotene, and it is followed by second stage of prophase. In the previous NCERT textbook, it is given as bouquet arrangement. The nucleus will be having bouquet arrangement. So this we call it as bouquet arrangement. So like this arrangement will be there. And during this phase of zygotene, the pairing of homologous chromosomes takes place. So the homologous chromosomes, which is one is from father and one is from mother. So I'll change the color. So the pairing of homologous chromosomes takes place. So one is maternal one and other one is paternal one. Okay. So this we call it as synapses. And this complex altogether with proteins we call it as synaptonemal complex. Let's see. During this stage, the chromosome starts pairing together. by the process we call it as synapses. Such pairing of chromosomes, homologous chromosomes, the electron micrograph is the stage that indicates the chromosome. So, which it is indicated by electron micrograph. That chromosomes is accompanied by a complex structure which call it as synaptonemal complex, and we call this chromosomes as bivalent or tetrad. Bivalent is based on the number of centromeres. We have 2 centromeres. Tetrad is based on the number of chromatids. You have 4 chromatids. That's why it is tetrad. Bivalent is based on the number of centromeres, which means 2 chromosomes. Based on number of chromosomes, it is bivalent. Based on the number of chromatids, it is tetrad. This stage will be characteristic. This first 2 stages of prophase are very short-lived when compared to next. Next, packetine. In this packetine, the 4 chromatids of each bivalent will become distinct and clearly appears as tetrad. This stage is characterized by the presence of recombinant nodules. So you will be having the recombinant nodules, right? So the site of which the crossing over occurs between non-sister chromatids. The relationship between this chromatid and this chromatid we call it as non-sister chromatids. The relationship between this and this is sister chromatids. The relationship between this and this is non-sister chromatids. And the crossing over. So this is the point of crossing over. Means overlapping of one chromatid on the other. So crossing over leads to exchange of genetic material between the 2 homologous chromosomes. So the crossing over is actually enzyme mediated. So the enzyme involved in this process we call it as recombinase. So crossing over leads to recombination of genetic material between 2 chromosomes. And the recombination between homologous chromosomes will be completed by the end of packetine. Means the chromatids got exchanged. Only the chromosomes are linked at the sites, crossing over sites. So the beginning of diplotin next. So here what it will happen is here what it is going to take place is the synaptonemal complex is getting dissolution. So tendency to be recombined the homologous chromosomes. So tendency of the recombined homologous chromosomes will separate, bivalents separate to each other except at the sites of crossing over. So you can see here except at the sites. Except at this sites it moved. So these X-shaped chromosomes are called chiasmata because in Greek X means chi. So in oocytes, mainly in some vertebra, this diplotin can last for months and years, even if you take for humans. So our oocytes will last for months and years, will be staying at diplotin only. So final stage of meiotic we call it as diakinesis. Nothing but this breaking of chiasmata will happen and fully condensed, the chromatin material is fully condensed as chromosomes, and meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus will disappear and the nuclear membrane will breaks, and diakinesis is transition to metaphase. First thing, metaphase. Now the bivalent chromosomes align in an equatorial plate. You will be having 2 equatorial plates. The microtubules forms the opposite poles of the spindle attaches to the kinetochore of homologous chromosomes. If you see in this picture, Here the centriole is there. Here the centrosomes, two centrosomes. Here you will be having the spindle fibers. The spindle fibers get attached to the kinetochore of the chromosomes. So here you can see this two metaphyseal plates. Anaphase 1. The homologous chromosomes will separate while the sister chromatids remain attached to the centromere. Here what it is going to take place is only the chromosomes are getting separated. The chromatids are not splitting. The nuclear membrane, nucleolus will again reappear. If you can see in this, nuclear membrane, nucleolus again will reappear. And this is called as dyad of cells. So although in many cases of chromosome undergo some dispersion, they do not reach the extremely ended state of interphase nucleus. So this two mitotic division, we call it as interkinesis, and generally they are short-lived. And there is no replication of DNA during interkinesis. And interkinesis is followed by prophase 2, which is very simpler prophase than the prophase 1, which is very similar also, if you see. So coming to the prophase 2, meiosis is initiated immediately after cytokinesis, meiosis 2. So usually before chromosomes, they have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes will again become compact. At this stage, the chromosomes align at the equator and microtubules from the opposite poles of the spindle get attached. So here, what it is going to take place? So it is converting to chromosomes. Okay, nuclear membrane is dissoluting. So in this phase, what it is going to take place? The center alignment of chromosomes. And here we have the centromere and the spindle fibers are getting attached. Right? And attach it to the sister chromatids. Anaphase. Simultaneously, the splitting of centromere is going to take place. The splitting of centromere is going to take place. Right? So allowing them to move towards the pole by shortening of microtubules attached to the kinetochore. So telophase, meiosis ends with telophase 2, in which two groups of chromosomes once again get attached by the nuclear membrane. Cytokinesis follows, resulting in the formation of tetrad of cells, which means that is 4 haploid daughter cells. So significance of meiosis is mainly for evolutionary point of view, and the gametes are formed for sexually reproducing organisms. So meiosis will only happen in sexually reproducing organisms. Meiosis is the mechanism by which conservation of specific chromosomal number of species is achieved across the generation in sexually reproducing organisms. Even though the process paradoxically resulting in reduction of chromosomal number by half, it is increasing the genetical variability in population from one generation to next generation. Variation is very important process for evolution. This is about cell cycle and cell division. So you can see this is prophase. This is mitotic prophase and this is meiotic prophase. So meiotic 2 prophase, which is very similar. You can see the spindle fibers is moving to the opposite poles and it is getting attached. And finally the middle alignment takes place.